raising their hand, so please interrupt me as we go, because otherwise I won't realize. All right, so let's get going with the meta-analytic part. So what we are covering today, as Raya already mentioned, we are covering the systematic review part and then the meta-analytic part. Let's start by kind of making an inference so that I understand also a bit how we all, how we understand this, the importance of a meta-analysis. So let's imagine we have 9 pieces of evidence. In this case, we have 5 pieces of evidence that indicate a positive effect, and they are statistically significant. That's why I put them in blue. There are 3 pieces of evidence that are statistically non-significant. They might be a bit positive or a bit negative. And then we have a piece of evidence that is statistically significantly negative, so showing a negative effect of whatever we are looking at. All right. Intuitively, one could consider that the way of deciding if there is a positive or a negative effect is by casting a democratic vote in which the majority wins. This is what we call vote counting, which we very much recommend not doing. The reason is that different pieces of evidence might be more or less trustable. So they might be more or less reliable. So what I did here is just to change the size of these dots to reflect that actually for this specific data set of evidence with 9 pieces of evidence, the ones that found statistically non-significant effects were the larger ones, the ones that had a higher precision. And precision often is related to the number of individuals that we are measuring or the number of trials that we are running. So if you ignore precision, you will have to do it at your peril, because the answers by ignoring precision can be very different. All right, so what is a meta-analysis? In short, a meta-analysis is simply a statistical tool, actually. It is often preceded by a systematic literature review, which is what Sreya covered, and it is very important. It's a very important process if you want your meta-analysis to actually provide unbiased and robust results. But the meta-analysis itself is just a way of quantitatively combining all the evidence that you found through your systematic literature review. And one of the important things that make a meta-analysis really cool is that you can weight each piece of evidence by how much you trust this evidence. There are different ways of doing that, but one that is the commonly used and the one that in most cases should be used by, per default is to weight the evidence by the precision of the estimates, the expected precision of the estimates, which is a measure that is heavily related to sample size. So the goals of a meta-analysis, I've divided them in 3. The first, and perhaps the one that most people think of, is that we use meta-analysis to test overall effects. So to quantitatively calculate a number that is in support or in disagreement with the overall hypothesis that you had. But very importantly, and often ignored, we also want to try and understand the generality of whatever overall effect that you find. This is a concept that is heavily influenced by heterogeneity. Heterogeneity, I will cover it quickly later on, but in principle, heterogeneity is giving us an estimate of how applicable the overall result that we found is across contexts, yeah? And the surprising thing is that despite that heterogeneity can be very high sometimes, which means that generality goes down, is not always reported. And in many, many cases, in the majority of the cases, I would argue, is not interpreted. And this is very important to keep in mind if you want your meta-analytic results to actually be meaningful. The second main goal of a meta-analysis is to try and understand patterns of effects. And this is tightly linked to the concept of heterogeneity, because whenever you find heterogeneity in your data set, in principle, is something that is not negative per se, that your general result, your overall result is not general. It's not negative because what you can do next is to try and understand why there are different studies that lead to different results. And you can do that by running meta-regressions. So for example, what you might be interested in understanding is if there is a factor, in this case, on whether one effect size is published or not, that might explain why you find such a cloud of data points in your data set. So in this case, we run a meta-regression to compare published or effect sizes coming from published literature against effect sizes coming from other sources. And what we found is that there seemed to be a big difference in these effect sizes, to the point that the conclusions based on one type or the other would differ substantially. And the last thing that you can use meta-analysis for, even though it's rarely used, is that you can actually generate hypotheses, because you have this zoom-out view of the literature, and you might be able to test hypotheses that at the primary study level would require a huge amount of work and sample sizes. So for example, you may have a question in which you can, by compiling data from different sources with different characteristics, be able to not having to run an experiment with 10 by 10 factor combinations. All right, so one of the reasons why meta-analysis is considered an important tool and is often used is because year after year, we have an overwhelming amount of science that is being produced, and this amount of science is increasing. That's probably why at some point in the 70s, somebody thought that maybe it's a good time to start trying to find a way of summarizing quantitatively all this information. And that's where the first meta-analysis, or at least the term was suggested. And since then, the amount of meta-analysis have increased exponentially. Actually, they've increased faster than the amount of evidence we are producing, to the point that around 6%, a very rough number, it could be 3%, it might be 10, but let's say around 6% of all studies that we publish these days are meta-analysis. Again, one of the reasons is that meta-analysis are considered to be really powerful. And one very cool thing of a meta-analysis is that you can combine small studies or studies with low precision, if I want to go back with the term that I introduced just before, put them together and trying to understand if, despite these apparent conflicts that you might see in this picture, the overall effect suggests otherwise. And this is a really powerful feature of meta-analysis. They can help resolve what we, as readers of the primary literature, might consider actual conflicts in the literature. The way they do that is by actually weighting each evidence by their precision. All right, so how do we combine evidence? So using a similar scheme that Sreya used for her presentation, we could divide the process in how hard it is. The entire process requires many steps. And what is important to notice here is that the steps are hierarchical. The decisions you make at the beginning of the process are the most important ones for the end product. Yeah. So choosing an appropriate effect size and the moderators, this is very important. And it has to be done at the beginning when you are thinking about how to even perform the systematic review. so that you have in mind what to expect and whether you will be able to meta-analyze whatever you find. Here, maybe I would like to suggest, and I think Sreya would agree with me, that perhaps the hardest part of performing a systematic review and meta-analysis is to actually extract the data from the studies that you eventually find as valid for your meta-analysis. The reason is that the literature is extremely differentially reported, and it's quite a process to try and understand what people have done in each study, which is key for you to be able to extract these effect sizes eventually. And what you will probably find in most cases is that there is such a lack of information that you won't be able to extract all the evidence for the hypothesis you are trying to quantitatively summarize. So how do we combine the evidence? So we need a common currency. This is very important to understand. We cannot just simply take numbers from one paper to another and run means of those numbers. We need that all those numbers, all those quantitative results from the different sources are in the same currency so that we can compare them. And that's why we need to use different effect sizes or effect size types depending on your question. Yeah. So for example, if you are trying to analyze an experimental design in which there are two groups that are being compared, let's say the control and the experimental group, and you want to understand if the experimental group shows a higher value for whatever thing you are interested in, you are going to be using effect sizes that compare means, which are, by the way, the most common ones in many fields. So you might be using effect sizes such as Cohen's-d or HSGs, which are maybe names that you've heard before, or response ratios, which are basically simply a ratio of these means. There might be other questions for which you might be interested in understanding an association between two traits. So this is rather common in ecology and evolution, in which we look at association between traits whenever we cannot do experiments. And what you need to make sure is that you have access to these correlations from different studies. That might require you to find a reported correlation or to extract the data from a scatter plot like this one. Again, mean differences, there are different ways of doing this, but if you are interested in mean difference, what you normally want to know is if overall the mean of one group is larger than the other. And super interesting, you can also consider testing for a difference in the variance, in the variability of each of these groups, if this is something that makes sense for the hypothesis you are testing. And I just left some references there because it's a technique that it hasn't been used as much and is still quite in development. And then there might be, and for some of you this might happen, there might be cases in which you cannot use the standardized common effect sizes that everybody uses as Cohen's-d, HSG correlations, but you may have to use special or custom effect sizes because maybe the question that you have at hand uses a very particular way of, or very particular statistical analysis that is now easily reduced in one of the common standardized effect sizes. So for example, here, I'm just showing an example in which we looked at heritabilities, which is a value that comes from mixed models. So it's a variance decomposition model. Or in this one, we, rather than looking at differences in variance, what we actually looked is at variance levels across many different studies. And these things are possible. They may just require some thinking and some testing and trial because most meta-analytic models are thought for the common ways of doing or testing meta-analytic effects, which is the HSG, log response ratios, odds ratios, and correlations, mostly. All right, here is a way of kind of thinking differently, something that Shreya put together, like a little tree to decide what type of effect size you might be using. And this is something really very important to keep in mind before you even start screening your literature, because you need to decide if at the end of the day, you will actually be able to quantitatively combine all the information you find. All right. The funnel plot. I've decided to dedicate a few minutes talking about the funnel plot, which is a type of funnel, a type of plot that you may have heard of. You may have seen it in several meta-analyses in your field, and you may have seen that people often refer to publication bias test and funnel plots together. Yeah. So what is a funnel plot? All right, so a funnel plot looks like a funnel. Simple, straightforward. So what am I representing here? I have in the y-axis an effect size that in this case, I decided to call it a correlation, an R. Let's say it's a correlation coefficient. In the x-axis of a funnel plot, you are going to have a measure of precision. This can be sample size, which is the one I am using here because it's straightforward to understand, but sometimes it might be shown as precision. It might be shown as the standard error, etc. At the end of the day, it's just a measure of precision or of trust that you have on each of the data points in your funnel plot. And it's well correlated with sample size. It doesn't need to be perfectly correlated. That depends on the effect size you are having, but it will be very well correlated. So what you see here is that there are almost 1,200 data points in this specific funnel plot. What you have is that on the y-axis is the size of the correlation between the two imaginary traits, because this is simulated data. So you have, do you see my pointer? I hope so. Well, you have that for small sample sizes. So let's say for sample sizes of 4, even though we know for this simulated data that the overall effect for the entire population of effect sizes is positive, as you can see here, and it's around 0.2, let's say. At small sample sizes, what we observe is that there is high variance between the different estimates of the true overall effect. So what the funnel plot is showing you is how just by differences, or just by how different sample sizes will make your evidence more or less trustable. As you increase the sample size you use to generate this piece of evidence, this quantitative result, in this case, a correlation, the likelihood that you are close to the true effect, whatever that is much, much higher. And the shape of this association is a funnel. So the precision or the variability around the overall effect reduces heavily as soon as you increase the sample size a little bit. I don't know if that's more or less clear. I hope it is. This is very key for understanding how meta-analysis actually deal or handle these differences in precision to when they are weighting the evidence. Yeah. So what we have here is that, and this is very important, as sample size increases in this funnel plot, in principle, the evidence should not change. So the average in this side of the funnel plot should be the same as in this side of the funnel plot. The difference is that the variance around the average effect is smaller here in the right-hand side than on the left-hand side. This is key, yeah? All right. This is simulated data. You will rarely see a funnel plot that looks so much or so beautiful, actually, like this with this perfectly perfect shape. Most of the time, particularly if you have a lot of data points in your meta-analytic data set, what you will see is that there are like data points here and there. There is what we call heterogeneity, which is a concept that I already introduced before. So heterogeneity, basically, in short, is all the variance that we observe in our data set, meta-analytic data set, that is not explained simply by differences in sample size. So if we go back, what we see is that in this specific data set, all the differences that you see between the data points in this one are simply because of different sample sizes being used across data points. Is the variability that you observe simply because there is chance in whether you are hitting the true overall effect, or you are a little bit higher or a little bit lower? And heterogeneity is that additional variability that is not explained only by those differences in sample size. All right. So heterogeneity is actually the variance in the true effects. And heterogeneity is very important because if you don't account for it, if you don't interpret it, you will probably be making your result look more confident than they truly are. In addition, heterogeneity is very important because it allows you to ask additional questions to your data set. It's the way you will run additional analysis, often in the shape of a meta-regression, to try and understand why is there this additional variance that is not explained simply by differences in sample size. I'm hoping this concept is clear. If not, please just interrupt me. I'm actually gonna pass through this because I think it's a lot of numbers that we don't need. But essentially, what I would recommend, and this is my tip for whatever you are doing a meta-analysis, is that you embrace this heterogeneity and first of all, report it, and second of all, you interpret it. This can be done these days very easily in most software because they allow you to calculate what we call the prediction intervals, which are intervals that are often much wider than the confidence intervals, and they are much wider because they incorporate heterogeneity. And the power of those is that they allow us to predict in which range we should expect a new study similar to the ones that we just meta-analyzed to be placed, which can be really powerful to understand later on if you want to perform another primary study, or simply to understand the context dependency of the effects. All right. Let me just give an example to try and make it a bit more practical, this concept of heterogeneity. I'm just going to show the results of a meta-analysis that was published more than 10 years ago, in which they had, let's say, a simple question. Yeah, they were trying to understand if females in birds, if they lay eggs of different size, depending on whether there is a male or a female chick inside. I know for the non-ecology evolution people, this probably sounds quite odd, but yeah, this is the sort of questions we evolutionary ecologists try to understand. In this specific meta-analysis, what they found is that, as you can see, the overall effect, this black thick line, was very close to zero for their effect size of interest. What they also observed is that for this specific meta-analytic data set, heterogeneity, and this is just a way of measuring heterogeneity, there are many, heterogeneity was quite low. So what they could conclude in this meta-analysis is that not only there is not a clear association between these two variables, So the answer to the question would be no. Not only that, but actually, once you account for differences in sample sizes between all these pieces of evidence, it seems that there is actually a generality in the fact that this hypothesis does not seem to be true. And this is something you can see visually in this plot by observing that there are only very few data points that are outside the predicted funnel shape of this funnel plot. If for this question, the funnel plot would have looked like this, where we have data points all over the place and a high heterogeneity, the conclusion to, or the answer to the question, do females adjust egg size depending on offspring sex, would be no, but it seems that there are many cases in which the answer to the question is true. So for example, the ones on top here, maybe for some species, the answer is true. And actually in this specific funnel plot or data set, for some data points, the answer is the opposite of what was expected. And these large sample sizes are not related to, these large effects are not associated with the different differential precision in the estimates. Okay, I hope I more or less explained the concept of heterogeneity. The reason why I also wanted to consider or wanted to spend a few minutes talking about the funnel plot is that because the funnel plot is key for how we test for one of the most important sources or one of the most important types of publication bias, which is what we call small study effects, which you may have heard of. A small study effects is a type of publication bias that normally is present or shows up by what you have is that you've collected your meta-analytic data set, and what you notice is that there are certain effects that are small in size, so low precision, low sample size, that are often statistically non-significant, that do not seem to be in your funnel plot. So your funnel plot, if you look at this, it looks like it has some gaps associated with small sampled effect sizes that are statistically non-significant. This, of course, assessing this visually is really tricky because, again, this funnel plot is actually very nice looking despite the random heterogeneity that I added. But this is more or less what you want to understand to see if there is small study effects. And the way you do that is by testing if the evidence, the overall effect that you find in your meta-analysis, changes across sample size. So when there are small study effects, what you observe statistically, not visually, statistically, is that as sample size increases, so you go to the right, the evidence here represented as the red line for the hypothesis you are testing with your meta-analysis decreases. This is very much suggesting that there is something going on in your compilation of evidence, and specifically suggesting that you are probably missing out on some effect sizes of small size and based also on small sample size. All right. So publication bias, now that we've introduced the topic, occurs whenever the effect size is included in your meta-analysis, generate a different conclusion to what an all-inclusive meta-analysis would have reached, assuming that's actually something possible. The issue with publication bias is that if you have publication bias in your data set, which is something rather common, this distorts the conclusions you can obtain from the meta-analysis unless you are very aware of it and interpret this publication bias evidence. So the surprise here is that, well, not all studies are published. So it's not only that it's difficult to find studies, it's that some of them might not be available because they were never published. There are many different reasons for why some studies might not be published or not. We often think of one reason as being one of the most important, and is whether the study found or not statistically significant results. But there are also other reasons for why the studies might not be available to you when you are performing your search, your literature search. There are also many different types of publication bias. So the studies might not be published, but there are other biases related to whether the studies are easily or are more easily or more difficult to found. Some studies might be published later. Some studies might be more difficult to access because they are published in journals that you may not have an easy access to these studies. So for example, when we discussed with Shreya these biases about language, not searching for different languages when you are searching for literature could lead you to not being aware of literature that is published in other languages than English. So for example, literature that might be published in national journals, which might contain, more often than not, studies that show statistically non-significant results. And this could be generating inadvertently a perceived problem in how a perceived publication bias problem. So this is also something we need to keep in mind when we are searching for literature. All right, how are we doing with time? I can see some questions in the chat. Is this something I have to deal with, or?
I am responding to them, but we can come back to them later.
All right. So is there evidence of publication bias in our literature? I would argue there is a lot. And to be honest, I think in many cases, in the majority of the cases, you probably find at least some evidence that there might be some publication bias in your meta-analytic data set. So this is a study I wanted to show, which is basically a meta-analysis in which we did an assessment of a quantitative assessment of meta-analysis published in Ecology and Evolution to try and understand if overall, across meta-analysis, there seemed to be evidence for small study effects. So small study effects, remember again, they show up whenever what you find is that evidence for the hypothesis you are testing decreases as sample size or precision increases. And what we found is that indeed, when you look at across meta-analysis and you quantitatively summarize these asymmetries in the different funnel plots, there seem to be, again, evidence overall for a negative relationship between effect sizes, evidence for the hypothesis, and precision of the evidence. There is another type of publication bias that you should be aware of, even though it's a bit less common, that is called decline effects. This is a type of publication bias that basically shows up whenever you observe patterns of declining evidence over time. This can be for many reasons. One of the reasons could be that the first original estimates of the hypothesis you are testing might have been errors, perhaps some of these data points in the funnel plot that might not be true. And as people start testing this hypothesis more and more, what happens is that the evidence declines over time. So this you can test simply by having access to the year of publication of or the year of publication or generation of the evidence that you are meta-analyzing. And this is also a simple test that you can do to ensure that you don't have these sort of patterns in your data set that could suggest publication bias, okay? And for this 87 meta-analysis, we also found evidence suggesting that even though there is context dependency, there is heterogeneity, overall, there seemed to be evidence for decline effects across meta-analysis. The important thing is that once you account for these biases, the overall effects, so the magnitude of these different effects across meta-analysis, can be heavily reduced. In this case, our study suggests that the reduction would be at 25%, which is a considerable adjustment. And this adjustment is actually something you can do at your meta-analytic level. Okay. All right. How can we do it? Well, I'm going to pass this. So here is a method that we suggested for how to test and adjust for publication bias in your meta-analysis. Even though there it says for ecology and evolutionary meta-analysis, the method is applicable across disciplines. The idea is that you are running a meta-regression to try and understand if there is this pattern of declining evidence as sample size increases. And the cool thing of this method is that by looking at the intercept of these meta-regressions, you can have at least a sense of what the overall effect of your meta-analysis would look like after you adjust for that decline in evidence as sample size increases. And this is something we explained there. We also have some workshops that are on YouTube, as well as some materials that I recommend. The important thing here is that if you run a meta-analysis and you don't test for publication bias, different types of it, chances are your conclusions are not robust and reliable. And this is very important. And this seems obvious, but there are many, many meta-analyses out there that do not report having tested for publication bias. And the amount of them that actually interpret the publication bias test is much, much lower, which is a bit worrying because chances are that a lot of the conclusions that we are writing in our meta-analysis are likely oversold. All right. I'm gonna jump on this so that we leave questions for the. Okay, so I just wanted to go through some examples of some of the meta-analysis we've run, just to try and discuss how to interpret results, okay? Which is something I think is very important. So this is a potential plot of all the evidence that you may have included in a meta-analysis. So what you have here, this is an orchard plot, it's called, which is very informative because it gives you access to all the data points. It sorts them, and then it gives you the overall effect, the confidence intervals, which is what most people want to report to know if the mean is statistically different from 0 or not, and then the prediction intervals, which are not that often reported, and I would very much recommend reporting since they incorporate heterogeneity. So the first conclusion you could be tempted to say from this plot is that whatever hypothesis you're testing, Alfredo, there is no evidence for it because the overall effect is very close to 0. And the confidence intervals, which are the thick lines, are heavily or largely overlapping with 0. And this in principle is one way of interpreting this plot. What I want to make a point here about is that this plot also suggests that there are many cases in which after you account for differences in precision between all these data points, so differences in sample size, the hypothesis seems to be true for some data points. So for example, the ones beyond the prediction interval, and the opposite for some of them. And what this is suggesting is that the interpretation of this a priori statistically non-significant mean has to be much more nuanced, because there seems to be some context dependency in which the hypothesis is true. And now your job as a researcher would be to try and understand which contexts are actually leading to the hypothesis being true. And you can do that by running meta-regressions. In the meta-regressions, what you do is to try and explain the variance between these data points by using factors that can be categorical or continuous. I don't remember if I have an example here. Yeah. All right. Here I would repeat myself, so I'm not going to do it. All right. Maybe just to conclude, and then we leave time for questions. So meta-analysis are in principle a very powerful tool. Remember that the meta-analysis actually in itself is only a statistical tool that allows to quantitatively summarize information. But meta-analysis are powerful only if they are done properly. Before running the meta-analysis, there is a key step that is very often neglected or not well thought, which is the systematic review. And I hope that Sreya convinced you of how important it is to really keep track and think well about what you are doing to actually reach the data set that you are going to extract information from. And then at the meta-analytic level, there are many decisions that you will have to make before you actually analyze your data. So it's very important to think about what you are going to analyze. And once you have your results, what is very important is that you interpret them appropriately. You interpret them not in a reductionist way, in which you only focus on whether the mean is statistically significant or not, but also understanding what levels of heterogeneity you found, how do they change the interpretation of the overall effect, how do they provide information about potential context dependency. And very importantly, you also make sure to test for publication biases. You can test for different ones because we know that they are commonplace. So ignoring them will only make your overall conclusions more robust and less reliable. All right. Maybe just as a final thing, Sreya suggested them. I think there are many guidelines there for how to perform systematic reviews and meta-analysis. Well, actually, there are many guidelines for how to report systematic reviews and meta-analysis. There are also guidelines, and Sreya shared a few, for how to perform systematic reviews and meta-analysis. What I think is important is that you read these guidelines and keep them in mind before you actually start screening and extracting data. Because what tends to happen with many people is that it is only at the end of the project that they realize they have to fill in a Prisma checklist. And then it's too late, because you will realize that you missed a lot of important things. You will also understand that it's very hard to do a posteriori, because you actually don't fully understand what is being asked if you haven't thought about it well in advance. And my recommendation is that you really front load the projects before actually starting to think about the analysis. And I think we can leave it here. I've been talking to myself for like 45 minutes. I could not see anybody, so I hope you are still there. And I'm hoping you also have many questions that we can try to help you with now. I'm gonna stop sharing the screen so that I'm back to seeing you.